Hello, everyone. Today we'll be talking about microbial genetics in the context of the hair care example once again, and this is the sequel. We're going to be talking about a whole new set of interesting information in the context of hair care. So the learning goals for today is by the end of the lesson, I want you to be able to predict how bacteria can regulate gene expression in response to the environmental change. So for example, if there's hair oil on the scalp, and explain the operon model and catabolic repression and predict effects on the gene regulation. So these are the stickers for today's lesson. Again, these are just modeling the 5 E's. So the first one starting off with engage. I want to be able to engage you guys with the lesson and introducing this really new information in the way that's understandable. And then I want you to be able to explore. So we're going to be exploring these new concepts and these new topics. After we get a general understanding of that, I want you to be able to explain, not only me explain to you, but you explain to your group and others in the classroom. And this is really going to facilitate your understanding. And then once we get that level of understanding, we're going to be elaborating on what we know and being able to apply it to new topics and new types of questions. Then lastly, I'm going to evaluate what your understanding is and to make sure that you fully took away everything from the lesson that you're supposed to. So for today's lesson, again, we're going to be starting off with just predicting how bacteria can regulate gene expression in response to environmental change. And again, for this example, a hair oil on the scalp. So here's a story time. It's wash day and you're picking a shampoo. If your hair holds oil very well or it retains oil a lot, you might use a clarifying shampoo. If it dries out really fast, you might have to choose a moisturizing one. You're not the only one reacting to oils. Your bacteria are also on your scalp, like Cutibacterium acnes, and they're also reacting as well. When there's more oil, they turn on genes to make lipase, and it's a protein that breaks down the oil for fuel. When the oil is low, they turn those genes off to save the energy. And this is how bacteria regulate gene expression based on your environment. And I want you to take a couple minutes to yourself just to engage your prior knowledge before this. How might your shampoo choice affect what genes bacteria turn on or off? And you might not understand exactly what I mean by the different types of shampoo. So I included this picture to the right. And these are all the different variations. As you can see, there's moisturizing, clarifying, strengthening, restoring. Talk about your personal experience with shampoo that you use to wash your hair and how that might play a part into the bacteria. I'll give you a couple minutes. And then I'll ask for 5 hands. If you guys can share 5 hands, then we'll move on to the next part. So your shampoo actually changes the oil levels on your scalp, and bacteria like Cutibacterium acnes can detect that. Bacteria will respond to changes by turning on or off certain genes. And I want you to explore how bacteria know when to express a certain gene and using the operon model, which I'll go into a little bit more in depth later, and we'll be talking specifically about the lip operon as well as the lac operon. The lip operon controls the proteins that break down oils. So these are our guiding questions again. But the first one being, how do bacteria know when to turn certain genes on or off? How might using different shampoos affect bacteria on your scalp? And then how does the Operon model play a role in this? First and foremost, we're going with how do bacteria know when to turn genes on or off? So I want you to get out a piece of paper, and this is really going to help track your exploration into all this information and make sure you're retaining everything that's key and will be asked probably later on quizzes. And I want you to be able to define each word in your own words as we encounter them. So I'm not saying write word for word what's on the slide. Interpret what you think that it means to yourself and what's going to be easier for you to remember in the future. These are the vocab words I really want you to pay attention to. So operon, inducible operon, and we'll be talking about the lip operon, repressible operon, a repressor, and a reducer slash an inducer, and the lac operon. And then lastly, we're going to be talking about glucose. At the end of the lesson, we will write a short prediction and explanation. And this is really towards the end. But if someone stopped using hair oil and switched to a clarifying shampoo that removes oils, what might happen to gene expression in their scalp bacteria? I want you to use at least 3 vocab words within that explanation to that question. Again, don't worry about it right now, but I want you to include the words like lip operon, repressor, glucose, and reducer. So I'm going to be going over the regulation of bacterial gene expression, the operon model of gene expression, what an operon means and the two different types of operons, the pre-transcriptional control, so what repression looks like and what induction looks like in that case. And then after that, I'm going to have you guys take a minute and reference your stellar vocab. I want you to talk to your group for about 3 minutes and see if there's anything that you miss or you don't understand. And again, I should hear everybody talking during this time. If you have any questions, this is also a time to ask any questions to make sure that you understand the information. Now we're going to go into our second guiding question, which is how might using different shampoos actually affect bacteria on your scalp and how it expresses genes. So this is again going over once again the operon model, what it looks like, what it means, and the two different types. And then I'll be going over the inducible operon and the repressible operon and what both of those operon looks like in the context of oil and when the lip operon is being used in that example, and how the purpose of it and how it saves energy or if it actually prevents overproduction. And then now we're going to be doing one of my favorite in-class activities, which is the WISE activity. I will be giving them not an article, but a tweet that is falsely generated, but they're real people that could have possibly said this. We're going to be taking about 10 minutes to do this activity, and this is really our exploring and our explaining phases being put together. We're going to be using the WISE framework to be able to analyze if it's actually a reputable resource and if we should be paying attention to it. So we're going to be using the W, which is who is the author of the source. I, which is the intent. So, what are the main ideas that they're trying to convey? S is the sources of their claims, and E is elsewhere. So, do other reputable sources say the same thing? I want you to get out a piece of paper, talk to your group, and write down your answer. Your presenter for each for your tweet will be the person wearing the most brown in your group. So, for example, we have our first tweet here, and you're going to be going over the W, the I, the S, and E, writing out each part on your piece of paper. Group one will have this tweet, and I want you to give one piece of evidence to refute this claim. So, go do some elsewhere research, and I want you to cite it on your paper. Fill in the information, and you're going. Oh, sorry. If it's easier, I want everybody to be able to fill in the information on this slide. And I will be providing a link to the slide deck so you can put it on there, but also still make sure it's written down somewhere, just so you have your personal understanding of it. And then also when you're presenting it to the class, we can all get a better view. So this is our first one from Taylor Swift, which says, other hair oils can permanently rewrite the DNA of your scalp bacteria. These changes cause lasting effects on your hair and health. Shampoos must remove all oil to prevent dangerous mutations. My new Swifty shampoo will be in stores. This is another one, same directions by Dr. Ann Madden. She said scalp bacteria can sense hair oil and switch their genes on or off, like flipping on a light switch. They only make enzymes when they need to, saving energy with smart gene control. So give one piece of evidence to refute this claim or to support this claim, and then fill the information in the slide, and you will explain your thought process. And then the last one is from Aiden Ross, and it says all bacterial genes turn on only when needed. There's no such thing as a gene that's always on. Bacteria are too smart to waste energy like that. Stop believing big pharma and buying stupid stuff. And then you're gonna again give one piece of information to refute the claim, and then you're gonna present it to the class. And then these are your opportunities after that activity. Everybody's explaining, everybody's doing that thing. I think this is really interesting because these are also like relevant people, maybe, and they'll be able to really check their sense of evidence checking as well. So now I want to get into the second half of the lesson, which we're going to be talking about explaining the operon model and catabolic repression and predict effects on gene regulation. And a guiding question that's associated with that is how does the operon model help explain how bacteria respond to available nutrients like oils? So we've explored the lip and the lac operon and how they, sorry, you're right. We're about to explore how the lip and lac operon and how they help regulate enzymes. The lip operon controls the proteins that break down oils. Now, let's explore how bacteria know when to express these genes using the operon model and specifically the lip operon. But we also will be talking about another very common one that you're going to probably see for the rest of your biology career that's referred to as the lac operon. And this is another bacterial strategy, the lac operon, and it helps break down sugars like lactose. And just reemphasizing, we're going to be talking about the lac operon. The lac operon breaks down lactose only when lactose is present and glucose is low. Throughout the lesson, we'll have some similarities to lac versus lip. So I'm going to be going over the operon model and what the structure of that looks like. And then for the lip operon model, I'm going over the structure of that, and that's where and showcasing where the difference between the two are, which is really in its structural genes, and what an inducible operon looks like, an induction of the lac operon looks like. And if it says include video from lecture, there is a video from the original slide deck that I would like to include in this, but for some reason I have not been able to find out how to replicate it. I'm going to be using this for my like research. So I need to figure out how to download that video to include it in this part. And then we're going to be going over the repressible operon and then the repression of the TRP operon. And again, it's the same video that I need to figure out how to put inside of it. And then so now we're just going to be doing a little facilitated discussion portion just to make sure that we understand what we're talking about. So in the group, we're going to take turns explaining the operon model using the following prompt. So one person or two people are talking about what is an operon, one or two people are talking about what's the difference between an inducible operon and repressible operon. And I want you guys to use examples of the lip operon or the lac operon. And then why do bacteria use operons instead of turning genes on one at a time? After you shared together, I want you to write one sentence that summarizes what you've learned from this conversation just now versus before the conversation. I'm going to roll the dice, so have your sentence ready. And again, we're going to further go into more about this, but more specifically, focusing on the catabolic repression portion and how that can affect gene regulation. So at this point, we've explored the lip and lac operons, how they help regulate genes. And now we're going to explore how the lac operon does more than just switch on and off, but it uses positive regulation to fine tune expression based on the cell's energy needs. And we're going to explore how catabolic repression helps bacteria prioritize the best energy source. In this context, we're talking about the lac operon, like glucose, before switching to lactose. And as I mentioned, we were talking about positive regulation and what's that look like in the context of lac. Um, when glucose is present and lactose is present, which one it prefers. When lactose is present and glucose is scarce, what it does. And then the lac operon and the full presence of it and what it looks like. And then we're talking about catabolic repression, so how glucose and lactose compare over time, and the number of cells and how it uses glucose more efficiently. Same thing. At this point, we're towards the end of our lesson, so we're going to do a quick wrap up. We're going to get out those notes that we've been working on with the vocab and everything. We're going to write 2 to 3 sentence response to this prompt. And when we're done, we're going to discuss it with our group. The person wearing the most blue in this case will be our speaker. So describe how bacteria know when to turn genes on for using lactose, and what role does positive regulation play in this process? And at this point, everyone should have something written down because this is going to be a part of our exit ticket. And then after that, we're going to have the person wearing the most blue, they're going to be the presenter. And I'm going to go over in summary. So bacteria can turn genes on or off depending on the environment, like the amount of hair on the scalp. The operon model is used to explain how genes are grouped and controlled using switches like inducers and repressors. Catabolic repression is the process where bacteria chooses their best energy source first before turning on other genes. And the regulation of gene control helps bacteria save energy and adapt to changing environments. And this funny little comic that I found online, it says, our mistake, we've introduced politicians' genes in that one. So it's like the rats are protesting because they've introduced a different type of gene inside. And again, just that stellar vocab. You're going to be working on the short prediction that you have, and I want you again to use at least 3 vocab words in your answer, like lip operon, repressor, glucose, and reducer. And you have to have something written down because this also will be part of your exit ticket as well. So include those 2 pictures to your exit ticket, and then we'll be doing something called a 3-2-1 exit ticket because we were wrapping up a section of a lecture that has really works together very well. I want you to tell me what are 3 new ideas you learned over the course of this week. What are 2 things that you learned that you will tell others about, whether it's your family, your friends, your cousin, or your coworkers? What is one thing you will do to make sure that you understand the lesson better? And then include your pictures that I mentioned that you need to have included. And that is it. Thank you. Thank you so much.